Hello and welcome to Psyched, the show where we explore psychedelics through social, economic, and political perspectives. Let's introduce our next speaker. Next up, we have David Nichols. David Nichols graduated from the University of Iowa with a PhD in medicinal chemistry. He then was a postdoctoral fellow in the pharmacology department at Iowa, followed by researching and teaching medicinal chemistry and molecular pharmacology at Purdue University for almost 38 years. With government permission, he synthesized high-purity MDMA, DMT, and psilocybin for clinical studies. He has written over 300 scientific publications on psychedelics, dopamine agonists, and more. In 1993, he founded the Heffter Research Institute, which has supported and funded clinical research with psilocybin and led the psychedelic renaissance. He is currently an adjunct professor at the University of North Carolina, Chapel Hill. David, thank you so much for joining us and welcome to Psyched.
Good morning. Thank you. So this will be a little bit different. I'm going to talk just a little bit about science, but not too much. And I don't have enough time to really talk about this in any detail. So I'm going to give you just an overview of the chemistry kind of psychedelics with a little bit of pharmacology, but not too much. So the thing about psychedelics is they're very different from any other class of drug, and we all know that. And this definition is actually in kind of the pharmacologist's Bible, Goodman and Gilman's Pharmacological Basis of Therapeutics. The 7th and 8th editions said, psychedelics, the feature that distinguishes psychedelics from other classes of drugs is their capacity reliably to induce or compel states of altered perception, thought, and feeling that are not or cannot be experienced otherwise except in dreams or at times of religious exaltation. A very strange definition for a class of pharmacological agents, really unique compared with virtually any other type of pharmacological agent. Historically, we know that peyote has been used for about 5,000 years. Archaeological excavations have found dried peyote buttons in caves in the American Southwest, used as early as 3700 BCE, before the Common Era. It contains a simple molecule. It's a trimethoxy. This is a 3,4,5 trimethoxy phenethylamine. Its identification was completed really in 1897 by the German pharmacologist Arthur Heffter, who had a PhD in chemistry, a PhD in pharmacology, and also an MD degree. He extracted the alkaloids from the peyote cactus, took all of them, each of them himself separately, and identified mescaline as the active compound. It was synthesized in 1919, and so there was a lot of early work in the 20th century with dilettantes and artists and poets who were interested in the effects of the peyote cactus. Then we had the mushrooms in South America. These mushrooms are used by the Aztecs. They were known as teonanacatl, and these mushroom stones were all throughout South America. Spanish missionaries tried to destroy them all because they believed they were pagan worship, but there were some 200 or so that were recovered. And Gordon Wasson, a New York banker and amateur mycologist, read about them in this 1957 story, read about them, and then went to Mexico and took a trip with a photographer and his wife and met with Maria Sabina, who was the curandera who allowed him to participate in a mushroom ceremony. And that was the basis for the article in Life magazine that I showed in this previous slide. 1957 is when this was published, his story about traveling to Mexico, Huatla, and participating in the ceremony. And this actually is what Timothy Leary first saw that clued him into the use of psilocybin mushrooms. And Timothy Leary was a big proponent of psilocybin mushrooms before he got onto LSD later on. This is an example of the mushrooms. And they have a simple tryptamine. It's a 4 substitution here. It's a 4 substituted oxygenated tryptamine. When this R group is a phosphate, it's known as psilocybin. It's a very stable molecule. You can recrystallize it from boiling water. And in the body, phosphatases in the gut clip that phosphate off. And what you have left is an H here, an OH. And that's psilocin, which is the actual active species in the mushrooms that interacts with receptors in the brain. DMT is another classic psychedelic. This is the structure of DMT. You see it looks very much like a psilocin or psilocybin, except it doesn't have any substituent over here. It's just unsubstituted. In contrast to psilocybin, it's not active orally. So mescaline and psilocybin are both active orally. DMT is not active orally. It's generally smoked or insufflated. And it's found in the root of Mimosa hostilis. It's a crystalline material. And it has very powerful effects, but potency-wise, it's not terribly potent compared with something like LSD. What we've all heard a lot about is ayahuasca. And ayahuasca is really a mixture of DMT with alkaloids that prevent its breakdown. So if you would take DMT orally, what you would find, it would have no effect at all. It's broken down by enzymes called monoamine oxidases in the liver. So in the Amazon Valley, what they've done is taken a Psychoactive Ergolines. This is one example, also called chacruna. It has DMT in the leaves, and then Banisteriopsis caapi is this vine, and it produces alkaloids called beta-Carbolines. And what they do is bind to the monoamine oxidase in the liver, and it prevents the monoamine oxidase from breaking down the DMT. So the combination of the beta-Carbolines from Banisteriopsis caapi and Psychoactive Ergolines gives you ayahuasca. And here's an example. Here's the Banisteriopsis vines, which have been pounded down. It has kind of a red color. You put it in a big pot of water with Psychotria leaves, and they boil the dickens out of it and boil it down, filter out the wood and the leaves when they're done, and you get this thick brown preparation called ayahuasca. And it's essentially kind of an orally active version of DMT. And people who have taken both psilocybin and ayahuasca will basically say these are very similar preparations, ayahuasca and mushrooms. For the ergots, natural sources of ergots occur in a fungus that infects grain. This is a stalk of rye, and these black things are ergot fungus. They're called sclerotia. And the sclerotia are a sort of a hibernating form of the fungus, which contains a food source and the necessary components for this fungus to regenerate under the proper climate conditions. And so these ergot heads contain ergot alkaloids, produce ergot alkaloids. And they all basically look pretty much like this. So this is a typical ergot alkaloid, and it would have various kinds of things attached up at this position. And I'll show an example of that later on. But the ergot alkaloids all are comprised of these 4 rings hooked together. It's essentially a fairly planar structure, and they differ principally by what's attached up at this position. This is actually numbered as the 8 position. And so the various ergot alkaloids are characterized by what they have up here. So we have ergocristine, ergotamine, some others. So these are produced by the ergot fungus. And here's another example. So here's again the stalk of rye that's been infected with these ergot sclerotia, ergot grains here. And these actually are, like I said, a dormant form of the ergot fungus. If you take one of these little grains of ergot, the sclerotia, and you put it under conditions where it will actually germinate, this is what you get. So this is one of these little black grains. It's been magnified, but you can see it in here. And what these are is the fruiting bodies of the ergot coming out of that. So this actually is an ergot fungus, and typically, like all fungi, it has these fruiting bodies. And the alkaloids that ergots produce are fairly toxic, and I'll talk about that in a minute. Ergot will also, and this actually is Claviceps purpurea, which is the one that was classically around in the Middle Ages and throughout Europe. Claviceps paspali can also grow on grains. So this is a different strain of Claviceps. This is Claviceps purpurea, Claviceps paspali on tall fescue, or on Dallas grass. And in pasturage, these things occur in contaminated pastured grasses, and where sheep and cows ingest these in large quantities, they can actually become quite sick and can damage their organs. So ergot was a real problem in the Middle Ages. It was a scourge. They didn't really know much about it. And so when they had a year where the harvest was sort of damp and cold in the fall, ergot would tend to grow on the grain. And if you know the way they would harvest grain back then, they would take big sheafs of the grain and they would beat it on the ground to knock off the grain heads, and then they would collect the grain heads. And when this was infected with ergot, those grain heads would be in there, and so they would take it, grind it all up, and make flour out of it, and then eat the bread. And if they consumed it over a period of time, they would get ergotism. And ergotism has a number of symptoms. It can produce sores on the body, produce hallucinations, nervous spasms, burning sensations, temporary insanity. And what happens with these ergot alkaloids, the ones that are in the ergot fungus, is they cause a constriction of the blood vessels in the fingers and in the toes. So these are examples of gangrene in the hands and in fingers and toes. And this can become so serious, as in the case of the toes here, that these will actually die and drop off. And so people that suffered from ergotism, if they survived the bout of ergotism itself, would often be missing fingers and toes. This would become so necrotic, this gangrene would be so serious that they would just simply dry up and fall off. It was extremely painful and you had a burning sensation. So this is often called St. Anthony's fire. It was a burning sensation. And St. Anthony was a patron saint of people who suffered from ergotism. And again, here's an ergot alkaloid. Here's ergotamine. And so you see again this 4-ring bottom section, which is an ergoline. And when you add this piece up on here to the 8 position, this is ergotamine. So this would be typical of ergot alkaloids. This ergoline structure down here, coupled with some complex molecule up here, which would be characteristic of a particular ergot alkaloid. Ergots are also produced in the seeds of certain types of morning glories. So Turbina corymbosa was used by the Aztecs. It was called ololuki, and presumably they used it when they couldn't get the mushrooms. And ololuki has ergine or lysergamide. Again, you see the 4 rings here at the bottom. This is an ergoline ring system. And at this 8 position, what you have is just a simple imide substituent. So this is lysergic acid, amide, also called ergine or lysergamide. It occurs also in other species of morning glories, heavenly blue morning glories, for example, in the seeds, and Hawaiian baby wood rose. And these plants don't actually produce the ergot alkaloids. It's a symbiotic relationship of the plant with an ergot fungus, and it's the ergot fungus which produces the ergot alkaloids. Now, ergine is not very potent. It's about 1/10 the potency of LSD itself, but it doesn't have qualitative intoxicating properties that are very much like LSD. It's more of a sedative. Just to show you the similarity between the natural transmitter serotonin and LSD. Now this is again, notice the 4-ring structure at the bottom. This is an ergoline. It's a lysergic acid derivative. So lysergic acid is the ergoline ring system if it had a carboxylic acid up here. So this is the diethyl, these are ethyl groups, the diethylamide of lysergic acid. Lysergic acid, diethylamide. And serotonin is a 5-hydroxytryptamine, and you can see that it's very similar to the structure of the ergolines. And this discovery of serotonin actually being present in the brain in 1953 really was part of a revolution of neuroscience, because prior to this discovery, people thought that all the serotonin was in the gut and there was no awareness that neurochemistry played any role in behavior at all. It was all thought to be psychodynamic, poor parenting by the mother and the father, and children who had psychiatric disorders were suffering from poor parenting rather than some deficiency in their neurochemistry. So LSD was really part of the process in the early 1950s that led us to start focusing on the chemistry of the brain as being involved in behavior. Now the receptors that LSD and the psychedelics act on are called the 5-HT2A receptors. It's one class of serotonin receptor, and there are 14 different types. So this is one subtype, and this is expressed on cortical pyramidal cells in the cortex of the brain, mammalian brain. Any mammal has these receptors in the cortex. And these are expressed on these what are called apical dendrites of cortical pyramidal cells. These cells look like little pyramids. They're triangular shaped. You can see several of them here. And these dark areas are where the antibody for the receptor has been staining, and it shows the presence on these apical dendrites. And you can see the concentration of those receptors is very high. If we look at this heat map, low concentration going from purple to high concentrations to dark red. You can see the red represents where those receptors are located throughout the cortex. And they're located in layer 5 of the cortex. If we look at actually, this is a mouse somatosensory cortex. This is the outer surface of the brain. The dura would be out here in the human brain, for example. And the cortex is divided up into 6 layers, layers 1 through 6. And layer 5, right in here, is where those apical dendrites are. And these are the cortical pyramidal cells. And these cortical pyramidal cells are really critical to proper cognitive function, memory, behavior. Everything that we represent as unique beings derives from the action of these cortical pyramidal cells getting input from all the lower parts of the brain. So the blue are negative or inhibitory inputs, the red are excitatory inputs. And these are kind of like central processing units, if you will, if you want to draw that analogy, kind of like central processing units in the brain. And they're stacked parallel. So this is, your brain is essentially a pretty massive parallel processor loaded with these cortical pyramidal cells and all these cells that support their nutrition and survival and give them physical support. And how do we test these? Well, I've worked in this field since 1969, way before most other people did. I mean, I was sort of the beginning of this sort of a renaissance, if you want to call it that. And so to study how pharmacology works, or how a system of physiology works, you can use drugs that perturb that system. And probably most of the listeners have heard of Alexander Shulgin, Sasha Shulgin, kind of a famous rogue chemist on the West Coast. He passed away a few years ago. But he made a lot of these compounds, published 2 books, and they're worth getting if you don't have them. One is called Pihkal, P-I-H-K-A-L. The other is called Tikal, T-I-H-K-A-L. And they represent, the first one, Pihkal, is autobiographical. It's writing of him and his wife, Anne. And Sasha would make these compounds in his lab. He had a lab behind his house, and then would give them to Anne. Well, he would take them first, and if they seemed interesting, he would share it with his wife, Anne. And then if she liked it, they had a group of friends who would get together occasionally in another place, and they would all sample this compound. And Pihkal is actually a story. The first part is autobiographical, and the second part describes the synthesis and behavioral effects of 170 some phenethylamine type psychedelics. And Tikal is not autobiographical, but basically talks about the same description of tryptamines that he had made, diisopropyltryptamine and things like that. Well, we couldn't do that. We actually used an animal model. We trained animals to recognize the effects of different types of drugs. For example, we could train these rats to recognize the effect of LSD, and where we had 2 levers, one of these would deliver a food pellet, a 50 milligram sucrose pellet, which is like a little rat candy. And over 2 or 3 months, we could train these rats reliably to press only the drug that was associated with the administration, for example, of LSD. We used MDMA, mescaline, LSD. You can use amphetamine, methamphetamine, ketamine. Drugs can learn to recognize all these kinds of substances. And so if we'd train this rat to press on the right lever after giving him LSD, and we give him a new analog that we'd made, if he presses on the right lever, he's basically saying, I think you gave me LSD. And that was about as good as the models we could get. I should mention that as a graduate student and even a postdoc, I always had lots of people who volunteered to take compounds that I made, but of course as an academic, I couldn't do that. And just to show you an example, so this is LSD at the top, and we had rats trained to discriminate LSD from placebo. So here's the ED50. So this is the dose where half of the animals would respond on the LSD lever. So 45 nanomoles per kilogram of body weight in the rat. Here are 3 analogs we made where we locked the diethyl group. So if you hook this carbon to this carbon via the agency of a carbon atom, you can make a 4-membered ring. And these 2 methyl groups can then be on the same side of the ring, which is a cis-azetidine, or they can be on different sides, up and down or up and down. So you can have the SS or RR with respect to these methyls. And just to show you the SS, which actually appeared as a designer drug or a research chemical, as known as LSZ, you can see it has about the same potency as LSD, and in humans it has about the same potency. And the cis and the RR compounds drop off. So we use that a lot. All the work we did basically to characterize behavioral effects was pretty much done in this rat drug discrimination model. So in 2017, I was working in a lab that published the crystal structure of LSD and the serotonin 2B receptor, which is very similar to the serotonin 2A receptor. So here's a sort of a diagram of the actual crystal structure. And here's LSD bound in here. So this is the receptor. And this yellow piece here, I don't have time to talk about G protein coupled receptors, but this is a piece of the receptor that connects the top of helix 4 to the top of helix 5. So the G protein coupled receptors are 7 helical bundles packed into the neuronal membrane. And this is the piece that connects the top of 4 to the top of 5. It turns out that this occludes the binding site for LSD in the wild type receptor. So here's LSD bound in here. But actually, if you do experiments in a test tube in vitro in the laboratory, it takes about 3 or 4 hours for LSD to completely get associated with this receptor. And it takes as long as 10 hours for it to come back out. So this probably, at least partially, explains the high potency and long duration of action of LSD itself. It takes a long time to get into the 2A receptor, and it takes a much longer period of time to get back out. So it stays locked in that receptor. So low doses of LSD, 25 to 30 micrograms, are detectable. So I just want to mention the work that we've done sponsoring clinical research. Of course, when I started in this field in 1969, there was no clinical research. It all stopped in 1976, and the Controlled Substance Act of 1970 had placed psychedelics into Schedule 1, so nobody was doing any work in this field. And I got together with some colleagues in 1993, and I said, you know, it's really too bad that nobody's doing any clinical work. Why don't we find some philanthropists, get some money, and identify people who could do research in this area and sort of rejuvenate clinical research? So I founded the Heffter Research Institute in 1993, and this is Arthur Heffter. He's the one who isolated mescaline from the peyote cactus and identified it. And up to the present time, we've funded about $10 million to support research since 1993. And just to show you, we've done a lot of research. The first modern study of psilocybin treatment was done by Charles Grob at UCLA with advanced stage cancer patients. He published that in 2011. And then we had 2 papers, one from Johns Hopkins and one from New York University, both looking at cancer patients using psilocybin-assisted psychotherapy. Then Matt Johnson at Johns Hopkins published a study looking at nicotine addiction and treatment with psilocybin-assisted therapy. An alcohol study by Dr. Michael Bogenschutz, who's now at New York University. He published a pilot study showing that psilocybin-assisted therapy was useful in producing sobriety in alcohol use disorder patients. Both this study and the nicotine study of Matt Johnson's have been expanded. Matt's at Johns Hopkins, he's expanded this study. Michael Bogenschutz is at New York University. He's recruited now, I think, 96 or 97 patients out of the 100 we tried to get before the pandemic hit. And again, this is looking at psilocybin treatment of alcohol use disorder. My son, Charles Nichols at Louisiana State University, discovered that psychedelics are potent anti-inflammatories and also has looked at some of the effects in the brain. He has continuing studies. We actually set up a lab in Zurich. It's our Heffter Zurich lab that's supervised by Dr. Franz Vollenweider. And if you go to our website, heffter.org, heffter.org, we have a number of publications there that we've sponsored and supported coming out of not only Franz's lab, but other labs as well. And Dr. Peter Hendricks at University of Alabama is looking at psilocybin-assisted therapy for cocaine addiction, and that study is about halfway through. And then a number of studies at Johns Hopkins University. You know, the basis, all these studies laid the basis for them getting commitments for $17 million to establish a psychedelic research center at Johns Hopkins. So there are a lot of studies that are going on there. And so if psychedelics can treat all these different kinds of conditions, what's the mechanism? And this is what a a brain looks like normally. It's firing all the time. Here's the time in milliseconds. So this is real time. But this is the way your brain is firing all the time. And it generates electrical patterns. There's a network connectivity there. And Robin Card-Harris and his group at Imperial College have shown that under normal conditions, if you look at network connectivity, that is the patterns of that electrical connectivity in the brain, you see that there's not much cross communication between different areas in the brain. But when you take psilocybin, all the integration within these different areas and the segregation between other areas breaks down, and you get a global increase in connectivity. And this is sort of what happens when you take a psychedelic, psilocybin and LSD. And somehow there's a therapeutic effect that occurs when this happens. And then after the drug wears off, it collapses back into this healthy state of brain connectivity. And so as a consequence, what we're all looking at now and what most people are talking about is the role of psychedelics in sort of rejuvenating the field of psychiatry, which hasn't had any new drugs for many, many years. So I think I have to stop there, and I can take questions at any time now. Just to acknowledge graduate students and postdoctoral fellows, I was funded by the National Institute on Drug Abuse for 28 years and also had Robert C. and Charlotte P. Anderson Endowment. So thank you for your attention, and I'd be happy to answer any questions.
David, thank you so much. We really appreciate it. Incredible work. We've been following everything you've been doing for a while, so thank you so much for coming on. One of the questions from the audience is just asking about where they can kind of follow up with some of the things you chatted about. They're asking, is it possible to read somewhere about your drug distinguishing experiments design, specifically in rats? Or if you could tell a bit more about how it was done.
So most of my work has been published in the scientific literature. I did write a review called Psychedelics in 2016 for Annual Reviews of Pharmacology. It's a very comprehensive review of pretty much everything that was known in 2016 about psychedelics. If somebody sends me an email, I guess I could send them a copy of that reprint if they have no way to get it. But otherwise, it would have to be specific papers that we've published over the years. Some of the rigid analogs with LSD we published in 2002. So in the primary literature, I don't have any really any book chapter. All the book chapters I've written are all in academic type books.
Mm-hmm. The other question that I had personally was, there's a lot of talk in this space right now about certain demographics of individuals who shouldn't be interacting with psychedelics, people who are more predisposed to bipolar disorder or psychotic breaks of some sort. I was wondering if you could share a little bit about maybe the mechanistically how that might happen in the brain when an individual is more predisposed to those things. What's actually happening that can trigger that, if you are able to share? Yeah.
So all the clinical studies that we have done, there is a pretty rigorous medical pre-screening where we look not only at their current health, but consider whether they have any first-degree relatives that might suffer from schizophrenia, bipolar disorder, or whether they themselves do. That would be a disqualifier because we just don't know what would happen. Schizophrenia is a condition that usually besets people when they're in late adolescence or early 20s. And it's typically kids will go off to college and then they'll have a breakdown the first year. The stress of being new on campus, the stress will precipitate it. So actually schizophrenia starts developing probably in an early age. We have difficulty detecting it before about the age of 13 or 12, for example. But it progresses on. And so the age when people experiment with psychedelics is typically around that late adolescence, early 20s. So if you have somebody who is going to develop schizophrenia or some other illness, psychiatric disorder, a lot of times taking a psychedelic will catalyze the onset of that illness. There's no evidence, and studies have been done, there's no evidence that psychedelics will actually cause mental illness. But in people who are predisposed, it's possible it can catalyze that illness. We don't know enough about schizophrenia, bipolar disorder. These are kind of black boxes. And treatments have been discovered kind of empirically. So we don't really know why, for example, if someone was going to develop schizophrenia anyway and then took a hit of acid when they were 16 and then developed schizophrenia then, we don't know why that necessarily would catalyze that. But the current thinking is that they would have developed it anyway. It's just that experimentation with the psychedelic probably catalyzed earlier onset.
Yeah, thank you for that. The other question that I had was some of the investors that we have conversations with, some of the researchers and individuals who are really beginning to enter the space now, or maybe have been around through a philanthropic lens over the last decade, have become much more interested in being able to conduct this research without the need for animal trials, and really being able to kind of step away from that practice. I'd love to hear your thoughts on, does that really limit everything that we're able to do in this field? Have there been comparable solutions that have stepped into place to be able to allow for that transition? Or is that something we're really going to need for the next few decades to understand these compounds?
It depends on whether you want to be the first human to ever encounter a new drug that somebody's made. Most people, if you say, do you really want to be the first one to take this? Most people will say, well, it'd be nice if they gave it to some animals or gave it to somebody else. I couldn't use it because I don't have an MD, and so we were forced to use animal models. The drug discrimination data was very good in predicting. When we had done a lot of compounds in the drug discrimination trials, and it could basically predict the potency in humans pretty closely. But when you start talking about adverse effects, things that could happen that could go wrong, you really, in most cases, and the FDA has their Kefauver Amendment of 1962 said that drugs had to be safe and effective. So proving safety is something you have to do before you ever go into a human clinical trial. Now, Sasha, when he tested them on himself, would start with very tiny doses and work up to higher doses. And all the compounds he looked at were either phenethylamines related kind of to mescaline or related to tryptamines. So he felt he was pretty safe in trying those up at fairly high doses because they didn't have known toxicology or toxic effects. But if you came up with a brand new type of chemical entity, and this is being done now, the military is starting to look at this. If you find a brand new type of chemical entity that activates the serotonin 2A receptor and likely has psychedelic effects, there's probably no way that you could justify going directly into humans without doing a lot of toxicology testing to make sure that it didn't have some off-target effect and didn't cause seizures or some other serious adverse effect.
Okay. And last question, you did this really comprehensive paper on psychedelics back in 2016. I was wondering, today, it's been a few years since then. What are still some of the really big blind spots that we have in this field? What are some of the things that we really need to be investigating more, putting more resources into, and things that we just simply don't know about the field of psychedelic research?
If I were to do it today, the field that I would really go into would be medicine and psychiatry or biophysics. The brain imaging now that's developed, the brain imaging technology is really powerful, and these kinds of studies are appearing with increasing regularity. So psychedelics have proven to be a really powerful tool in studying consciousness. But we don't understand what causes depression, what causes anxiety disorders, what causes addiction. And I think the combination of psychedelics with brain imaging is going to lead us to a better understanding, and we can find different approaches. They may not even be psychedelics. Once we understand what actually happens that causes people to get, say, addicted to alcohol, once we understand the fundamental mechanism, we may be able to develop completely new treatments. So the psychedelics are really powerful in helping us understand how the brain works in consciousness, but they're also going to lead us, I think, to very new ideas and novel approaches in psychiatry for treating all kinds of disorders. And that's really, I think, that brain imaging technology coupled with clinical studies, I think, is going to really lead us to some powerful insights.
Mm-hmm. Definitely. Well, thank you so much, David. I know we didn't get to all of the audience questions, but thank you so much. And we're really looking forward to continuing the conversation.
Glad to help.
Yeah, be well.
You too. Bye-bye.